Welcome. I think the online, can everybody hear me? I don't have the chat up. Olivia, are you on online? Okay. Yep. Nick says we're good to go. It's been a journey. We're all here together. This is great. Yeah. So I've been asked to give a brief introductory remarks, sort of lay of the land. I'm going to dive right in. So first and foremost, good afternoon, everyone, and welcome to the AgBioData Annual Meeting 2026. My name is David Molik. I'm one of the organizers of this meeting, and I also serve as the department head of the Open Publishing Exchange and the head of New Prairie Press at Kansas State University Libraries. We're so happy to be supporting this. And we're really excited this year to be hosting at Kansas State Olathe campus. Lauren is here with the Olathe campus. We'll hold on for acknowledgements. And we're so happy to be able to bring in people to get both in person and virtually. And that hybrid model is intentional. It reflects a kind of distributed, collaborative infrastructure that I think AgBioData really prides itself on. And before we get into the substance of this meeting, I want to take a moment to recognize the people who are making this possible and who have made this possible. First, a huge thank you to Lauren Rickey here at Kansas State Olathe campus for event planning organization. This meeting doesn't happen without the level of coordination and care that you've provided. So thank you so much. I also want to recognize the organizing committee who have put in a tremendous amount of time in a very short period. These things, I want to emphasize this, these things usually take a year. They have done it in 4 months. Absolutely tremendous. And the amount of logistics and intellectual direction of this meeting that's really in that core group. So a special thanks to Marcela, Tello-Ruiz, Olivia Haley, Sarah Dreyer, who's hopefully online or hopefully actually asleep, Monica Poelchau, and Sunita Kamari. Thank you so much. And finally, thank you to our sponsors. Element Biosciences is here with us today. Thank you so much for supporting our endeavors. And IDEA, who is the digital agricultural group at Kansas State, has generously supported us as well. So thank you to them as well. Your support is critical, not just for this meeting, but for sustaining the broader ecosystem that AgBioData represents. So for emphasis, this isn't just another annual meeting. AgBioData sits at a really important moment. I think it's no surprise that databases, infrastructure, biocuration, and long-term sustainability of the data systems that modern agricultural and biological science are in a period of instability. And I want to emphasize that word sustainability, because what we're really talking about here is not just maintaining databases, but sustaining knowledge infrastructure. The people who curate and maintain it, the systems that store and serve it, and the governance models that allow it to persist beyond any one grant, institution, or funding cycle. It's hard, and it's getting harder, and hopefully this meeting can help us focus our efforts towards a better future. From the Kansas State perspective, hosting this meeting is a big deal for us. It aligns directly about how we're thinking about research infrastructure, integrating data, publishing, and repositories, supporting open and public access mandates by design, and building systems that work across institutional boundaries, and supporting digital agriculture. And AgBioData is exactly the kind of network that K-State wants to be in partnership with, not as a host, but as a long-term collaborator. So here's what we're here to do. Over the next few days, we're not here just to share updates. We're here to do real work. That includes thinking seriously about sustainability models, identifying funding pathways and leadership structures, exploring shared infrastructure and services, and asking some difficult but necessary questions about governance, equity, and independence. If we do this right, this meeting isn't just a checkpoint, it's a pivot point. So with that, I want to thank you all for being here. Whether you've traveled to Olathe or are you joining us remotely, your time, your expertise, and your willingness to engage in these conversations are what make this network real. I'm excited to see what we can build together in the next few days. Welcome to the AgBioData annual meeting. Next, Monica Poelchau with the United States Department of Agriculture, the National Agricultural Library will be talking to us about it.
Welcome, everyone. Thanks for coming. Thanks to David for the introduction. As he said, I'm Monica Poelchau. I'm a geneticist with the National Ag Library. I lead the i5K workspace at NAL, Insight Genomics Platform and Database. And the hat I'm wearing here is as a longtime member of the AgBioData Steering Committee and as a co-PI of our network. So I am very pleased to be providing a perspective sort of to ground all of our participants in how far we've come in the past 10 years. Because as David mentioned, we have a lot of work in the coming days. And so it makes sense for us to figure out or to reassess where we've come from. So what I was going to tell you today, tell you about today is essentially give a history of where we've been. Let's see if this works. Yes, it does. Good. Before I do that, I would still like to acknowledge first who has been contributing to this network. I'm going to be delivering a lot of slides on work that AgBioData has produced, but I am not really the primary person who's done that. A big shout out. Let's see, does the pointer work? I'm scared. Here. Okay. Yes. Big shout out to the AgBioData working groups who have put in a tremendous number of hours to move AgBioData forward. There's no way for me to actually put all your names on there. So if you're either in the audience or right here, a big shout out to you for producing all the work that you've done in the past 10 years. Our current program coordinator, Marcela Tello-Ruiz, thank you very much for being here and for moving us forward. We couldn't do what we do without our program coordinator, and also acknowledging our past program coordinators, Darwin and Anarita, and our steering committee members, the people sort of behind the scenes who make all this work. So acknowledging them first. Okay, so I wanted to give you a brief history of the AgBioData Consortium, where we started in 2015, then move on to our current work, which is the National Science Foundation Research Coordination Network grant that we have, and our progress on our 4 aims that I'll go into in a bit. So essentially the meat of the work that we do in our working groups, where we have grassroots definitions of the problems that we want to solve and implementation of them, expanding the network to include key stakeholders, education, and sustainability. And then that should all hopefully give us all sort of a shared understanding of where we are, because I see a lot of faces that I've known for about 10 years in this network, but also some new people here. So we need to get everyone on the same page. Okay, so a brief history of the AgBioData Consortium. There's the window over Lisa's face. Sorry about that. So I'm starting out in 2015, where when AgBioData was founded, and really we got going at PAG in 2015, where Lisa Harper, among a couple other people, but really Lisa was the founder, and with the main vision about AgBioData, that there were all these databases that we knew of: Maize GDB, Soybase, i5K Workspace, and we heard of each other, but we really didn't have that much of a connection point and ways to interact. Sorry, okay. Ways to interact and talk with each other. And so she had the vision of actually having a consortium and a regular way to meet and share common problems. So the goals that were defined at this meeting at PAG were to bring scientists together very simply in order to outline the biggest needs facing our databases today, to determine how to address these needs as a group, and to learn to work together, to leverage our work and have greater efficiencies among groups. That's the main message then, and I think that message still persists today about why AgBioData exists. So at that meeting, was anybody there at that meeting? Show this guy. We got about 5-6 hands here in the audience for those of you online. So there were over 70 people. I was looking back at the notes for this meeting and preparing for this talk, and I was like, wow, there are a lot of people. That was pretty amazing. I think we have about 70 registered for this meeting too. So we're holding strong with participation here. Representing 23 groups. Let me see if I can. that corner in my slides. Okay. In 2015, we already realized that we should have the working group model that we still use today, where we define issues that are important to our network, and we have volunteer-based small, virtual distributed groups that work on that. So our working groups at that point, just for historical interest here, were metadata collection, ontology use, data sharing, repository, search and discovery, UI, usability, and communication. Some of those still ring true today. We established a communication system. Anybody remember Basecamp? So I'm very glad we've moved on from Basecamp. It was nice, but now we're back on better track. And a regular webinar series that still persists today. So that's how we started, totally unfunded, based on Lisa's vision. At that point, we got going and started meeting regularly, and we realized that we actually needed to formalize our structure and have an in-person workshop where we really could focus in on the goals from the working groups and sort of codify AgBioData via publication. So we met in 2017, thanks to some National Science Foundation funding and Dory, Maine in Salt Lake City. Here are all of us looking 10 years younger who were there, and some other faces that we don't see here today. And that was a really productive meeting for us to actually that's an interesting thing that Google Slides is supposed to be an arrow that led to our publication on the AgBioData recommendations for sustainable GGB databases. And the groups that we worked on, the working groups that we had at that point ranged from like the entire spectrum of the things that you need to do as a database, from curation. A lot of what we do is curation of special data types, literature curation to establish gene function. At my database, we do structural gene curation, that sort of thing. Ontologies to structure the data, metadata and persistence to find it, database platforms to interact with it on software, programmatic access to data / APIs. Communication, of course, was always a big piece with us to figure out how to get the databases to continue to talk to each other, and sustainability, which we'll hear a lot about still in this workshop tomorrow. So at that point, we had our first white paper published, and we still realized that, a lot of the things that we talked about in that white paper were sort of a gap analysis of here's where the field of the land of our databases is, but no solutions. And we figured in order to have solutions for those problems that we were defining, we need to have more funding. And it took us 3 years, but we eventually were successful in getting a National Science Foundation Research Coordination Network grant that started in 2001 and ends this year. It was initially a 3-year grant, and we were very fortunate to receive a NSF creativity extension, which is actually a fairly rarely awarded extension for projects that were deemed of unique merit. So we're on our 5th year of funding now, and so I think now is a good time to take stock of where we are for the RCN. And here's the aims that we set out to do. How we decided to organize it after like the first 5 years of being together was, first, we had already codified this working group structure and the system where it seemed to be fairly effective for us to get together in topical groups and identify problems and work on solutions. So continuing to do that at a grassroots level, having people define what the problems are, and importantly, figuring out ways to implement solutions to those problems. And it's all around the scope of fair data, findable, accessible, interoperable, reusable. Our second aim was to very much essentially a network aim to expand the network to include key stakeholders, which are not just us database personnel, those managing it and running the day-to-day show, but our users, so stakeholders that are using the data, people who generate the data, publishers and funders. So essentially describing the data life cycle that we're part of as databases. Our third aim was based on the recognition that we can't really exist and continue to ask for funding if we don't define to our stakeholder groups or other researchers what fair data is and what databases are for. And so with that recognition, we set out to create a curriculum for researchers that could be easily provided to describe and train new researchers on what genetics, genomics, and breeding databases are and how we operate and why we help the data lifecycle. And finally, sustainability has always been a key problem. So how do we actually keep databases going, especially in a funding landscape where you have 5-year grant cycles, but we're really longer term projects that require maintenance? How do we actually make that happen and across as a network? So that's our fourth aim. So I wanted to now move on to describing how we've done on those 4 challenges, so 4 aims over the past year. So both progress and where we might need to still do some work in the last couple of months of our funding. Okay, so here's our nice roadmap. First, I'm going to talk about aim 1, so our grassroots-driven working group products. So when I was figuring out how to describe this and summarize it to everyone, we have a very, very great amount of working groups. So 16 working groups total, 12 of those from aim 1, 5 are active, 11 are completed, and they really run the gamut from all the different functions that a database can provide, and it was kind of hard to summarize. So I'll show you in a bit how I decided to do that. These have produced 10 peer-reviewed manuscripts so far as part of the grant. We had others previously as well, and 4 repositories with code or training materials. And so the first set of products that I wanted to describe are ones that essentially provided a gap analysis or a network-wide assessment of needs. And there are these 4 different categories here. The first is genotype-phenotype standardization. Essentially, that was a description or analysis of where the gaps are with respect to these data types. But they also produced a really great product where they assessed what data types all the databases that were currently in the network held, and that was actually then reused by, I think, the FAIR Scientific Literature Working Group. So we are able to actually reuse our own products. The next is the Data Federation and Ontologies Working Groups. Those were 2 that managed to also lay out network-wide assessments. The Data Sharing Working Group actually, as part of their next phase, they realized that education was a gap for our network, and so they actually provided training materials as a follow-on from their gap analysis. Data reuse was the next paper in 2025. And which essentially describes gaps in data reuse. An important point for data managers is that they actually, one recommendation that we might want to follow up on is to have standardized metadata across all the databases in the network. So there's some good recommendations there that could still be followed up on. And finally, the Phenotypic Data Management Working Group produced a paper on opinion piece essentially on the need for robust, fair phenomic databases. So essentially, these I'm showing you here because as we move forward in the next couple of days, it's good to remind ourselves what resources we have to draw on already that could be reused. The next set of stuff that I'm going to describe that is our products from our AIM 1 are the development of standards or technical solutions and training materials. So I'm going to show you 6 of those, and I'm going to show you one short example just to remind you of that one, which was pretty cool. So we have a GitHub Markdown resource on recommendations for pangenome analysis and archiving. We have a genome assembly and annotation nomenclature system and command line tool. Guidelines for genetic variation, harmonization using RSIDs, and I'm not going to talk about that because we're going to hear about that tomorrow. The products from the single-cell RNA-seq working group, namely curated plant cell data sets and the Shiny Piggy tool for porcine data sets. We'll hear about them tomorrow. Training materials from the Data Federation Working Group, which I mentioned before. They also exist as a GitHub Markdown set. And the Database Finder tool from the FAIR Scientific Literature Working Group for increased data discovery across databases. So I think, oh, actually, no. First, I'm going to dive in a little bit background on the GMOD identifiers. I didn't have time to go into detail about all these products, and I'm just sort of showing them to you. But this one, I wanted to bring some special attention to because I thought this one was actually, they're all cool, but this one actually was very, very close to my heart because I have always struggled with GMOD identifiers and codifying them and defining them, and also genome assembly identifiers too. And this working group recognized this and created a new system which you can see here, and which I will not go into gory detail on. You can read the paper to have a standardized a gene model identifier system that we can use. And I've actually reused this in our for the I5K workspace in some shape or form too. There is a code base that you can use to help generate these. And not just me, several other AgBioData members have adopted these or plan to adopt these. So it's a really cool example of sort of a very unglamorous yet completely necessary product that AgBioData is good for. Okay, so I'm going to attempt to do a poll. Let's see if it works. Hang on. Okay. Lauren, is Lauren? Oh, she's gone. Anybody know how to do a poll in Windows? Because I have it already set up. It's just. Never mind. Here, I will share again. We'll wing it. Okay. And I was going to record data here, so it's going to be kind of hard from the Zoom. Are you guys on Zoom? Marcela or Olivia, could you give me like a number of thumbs up for each of these questions? Cool, thanks. So, okay, you guys here in the audience. So I was going to ask, which of the following working group products have you used in some shape or form, just to remind ourselves of what exists and see what the uptake is? Because one of the things I wanted to see is, we have all these products. Do people actually know of them, and have they been reused? So for you guys in the audience here in person, could you just give me a hand if you've reused it? And I'll write this down here. And if you could, oh, cool. And for you in Zoom, if you could just give a thumbs up, is that the best way to do it? Let's do a thumbs up, yeah, and see if we can get some numbers here. Okay, so which of the following working group products have you used in some shape or form? So the database finder tool. Show of hands here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. Do we have a show of hands in the audience there? 2 online? Okay, cool. All right. Data Federation recommendations. Yeah. Nothing online. Right. Okay. Data Federation training materials. Oh, there was one. Okay. Okay. Data Federation training materials. Just me. Okay. Anything online? One online? Okay. Data reuse recommendations. Any online? One. Okay. Pangenomics. Oh, two. Okay, thanks. Pangenomics tool overview. One. I've used it. Anybody online? 3 online. Woo. Okay. Genome or gene nomenclature rules or software. Okay, more popular. 1, 2, 3, 4, 5, 6, 7, 8 here in the room. 2 online. Okay. 3 online. All right. Am I going too fast, online people? Sorry. Genotype to phenotype recommendations. 1, 2. 2 in person. 1 online. Okay. Ontology recommendations. 1, 2. Cool. 2. Phenotypic data standardization and management recommendations. That one's newer. 1 in person. 1. Okay. I think, oh, RSADs for SNPs. 2, 3. 3. Okay. I didn't, yeah, this is probably turning into a popularity contest. That was really my intent, but there we are. I'm new to AgBioData. I've just learned about these products. I wanted to accommodate that. 1, 2, 3, 4. Okay, cool. Track that from our account. Any new people online? No? Okay, cool. Yeah. Thank you for indulging me with that. I just wanted to like see what has been used and what has like been the more effective distribution mechanism for some of the resources that we produce, and that should be helpful for us moving forward too. Okay. Um, okay. So next, I wanted to go into our second aim, which is our sort of network aim. We wanted to expand the network to include key stakeholders. That was true over the entirety of our 5-year funding period, even after our 2-year extension. Acknowledgement that we did include sort of a diversity aim in aim 2, and we sort of had to dial back on that with how things have been going. But essentially, the aim has stayed the same, where we really need to ensure that we have a broad network that encompasses basically the data lifecycle for us. Okay. And so to show you some stats here, I adapted a figure from one of our recent papers where Anarita Morano is the lead author that's been submitted. And so our membership has increased. So we started out with, I guess, 70 or so, and now we have 389 members from primarily North America, but also distributed otherwise globally. We're 44 database resources. We might actually be more now. This was just when the figure was created, but that was a couple months ago. So, and we have, of course, primarily plant databases, some livestock, some insect here, and other database resources as well. We have these because of efforts that Anarita and the Outreach and Recruiting Working Group performed. So some of the outreach efforts were community workshops, so one of the ones that we're at today, conference workshops, primarily at PAG or the ASPB, and our webinar series, which still goes on today. And I'm sure we have more webinars than 32 right now. It's just an outdated slide. So it was a lot of work. If you've ever been to PAG and gone to our booth, you'd see that it was a fun experience. And that was another avenue for us to recruit more people. The Outreach and Recruiting Working Group was the main group that drove this focus, where the idea was to, as I said, recruit database stakeholders, including publishers and data generators. And we're pretty proud to have a fair amount of publishers actually hosting this conference. And new genetics, genomics, and breeding data resources to help implement the standards that we advocate for. This working group also had a webinar series, and I just wanted to do a brief shout out that they invited the 1890s land grant institutions national programs and also the SACNAS programs to come and talk to us to see what synergies there were between AgBioData and these groups. This was in the previous funding period. So to continue our recruitment and sort of network building efforts in this next period of the grant, we have some funding opportunities, which I hope you all are aware of, or if you're not, here they are. So the AgBioData Ambassador Program was what we proposed in our in the second funding period of the grant, where in response to sort of feedback from you all at one of our previous workshops, that it would be good to have another way to reward or acknowledge the efforts of all the working group members. So this is the Ambassador Program. If you're a current or past working group member willing to present about AgBioData at a conference, we still have awards of up to $2,000 for presenting work. So, and we do want to award these. So please apply. We love having the word spread. And here's the guidelines, or you're going to talk to us in the next 2 days. So give us a shout out if you're interested in those. Similarly, there's the AgBioData Championship Program, where the idea was, so we have the ambassadors to give talks, but the champions, we have a lot of work going on in the working groups, and this was intended to support people who are really providing a lot of effort in those working groups and taking on significant work. So these people would receive a $2,000 stipend for 1 year to advance the goals of the working groups. I think they write a blog post, right, and that sort of thing too. So, Oh, that's it. So if you didn't hear this online, Lenore points out that it could be less than a year if it's significant effort. So don't be shy about applying for this either if you are, I guess, and there's some certain stipulations. I think you have to be in the US and that sort of thing. Just to show you some examples of some of our previous fellows, our 2025 champions were Leila Kabugos from the Fair Scientific Literature Working Group, Marcela Tello-Ruiz, Standards for Genetic Variation, and Sunita Kumari from the single-cell RNA-seq biocuration working group. And our ambassadors for 2025, Jacqueline Campbell, Carson Andorf, Marcela, and Sunita again. So please do apply. We still have funds available for this year. All right, our third aim is around education and training. And again, the goal was to provide materials for our stakeholders or researchers that use our databases to understand why we're doing this. So yeah, the goal, create an educational curriculum around the history, importance, and best practices for fair data sciences in genomics, genetics, and breeding, and the history and structure of GGB databases and how they benefit the entire scientific community throughout the data lifecycle. This has a couple of modalities. So the focus was on fair data principles. The audience for this curriculum is educators of undergraduate and graduate students. So the curriculum is for the undergraduate and graduate students, and the people who would use that would be the educators. There are two modalities. One would be in person, so you could reuse the slides or adapt them for your own use. And the second is self-directed and asynchronous, so there are actually recordings of that curriculum online. It's open access, easy to use, customizable and remixable. You can see the different modules here on the right, from anywhere from what is a biological repository to databases for agriculture. You can see the reference there, thanks to Anarita and all the others from that working group to create that. And just here's just a screenshot of where you can find it, and then also Meg narrating how this the content of the curriculum there. So around this curriculum, one of the things that we wanted to do in our current funding period is to ensure that this curriculum is used and updated. So we have implementation or development awards for these, 5 awards up to $600 so 5 awards up to $600 each per year. And this is for if you are willing to teach it in your classroom, if you want to add new modules, if you want to translate that into other languages, that sort of thing. So we haven't had a lot of uptake on this award, and we need to figure out why. And we'd really like to make sure that this curriculum is reused. So for that, I have my next audience poll. I know you guys are super excited about this. So we could do the same thing. I have, I think, basically yes, no questions. So if you can give a thumbs up online for a yes, and then here, same thing, if you can do your hands for a yes, I guess. So have you used the education curriculum before? Oops. Anybody? 1, 2, okay. Anybody online? No? Okay. Are you in a position to use it? Who could use it? Anybody? 2? Okay. 2 here. Anybody online? No? Okay. That might be an issue. Are you interested in using it? 1, 2, 3, 4, 5. Okay. Anybody online? 1. Okay. Are you interested in being paid to use it? 1, 2, 3, 4, 5. Okay. Anybody online? 2 online? Okay. More getting paid. All right. Um, do you know anyone else who might be interested? 1, 2. I'm really trying to shout 3. Okay. 3, 2. Okay. 3 online. All right. So clearly, I'm trying to push for this to be taken up. So if you have thoughts on how we could improve the uptake or who to reach out to, please reach out to us. For example, for me, my thought was with this, I'm not an educator per se, but I do go to conferences, and there's often opportunities to teach workshops at conferences. Could it be adapted to that sort of format? PUI group at ASPB? Ah. Thank you, Doreen. Yeah, I think Doreen said to engage the P. Oh, you guys know because you saw the chat. Never mind. Okay. All right. Thanks. Moving on to the next slide. Okay. So our next aim is on sustainability of our databases. I'm not going to spend a huge amount of time on this because we're going to spend next afternoon on sustainability. So just sort of an overview. And some specific things. So here's a slide showing results from a survey that was conducted by Shabri from Phoenix Bioinformatics in 2022. And just to highlight here to show, can you guys see this even? It's one of the questions from that survey on database funding security. And the question was, how would you rate your current level of funding security for your database in the next 3 years? I thought that would be interesting to show right now with the different funding situation. And at that point, we have an N of 19 respondents here. 9 were in the moderately secure territory, and 10 were in the less secure territory. So be curious to see how things have changed. And some early recommendations from that survey were what was interesting is that there was recommendations on going back to the stakeholder community, especially regarding what's the buy-in for any of the sustainability strategies. The survey had several sort of sustainability strategies listed, so I'm not going to show those here, but what will be their buy-in and what's their perception of the value of your database? Like, is there potential for any of these, or is their perception of the value low? Another recommendation was to capture user statistics. I think the idea was to dial in on the user behavior and therefore streamline your resources to have the most effective set of tools that you can manage for your database, for your stakeholders. And then finally, there's a concern about data duplication across databases, and so to gain a better understanding of that. I saw some chats going in. Are there any questions from the audience, or are we good so far?
Says making it as a course and teaching as traditional course may not fly.
Oh, okay. That was some education. Yeah. Thanks, Sushma.
Also, Sushma, this might be used for conducting workshops offered through the library for graduate and undergraduate students.
Okay. Thank you, Sushma. All right. So just to give a brief overview of what the Sustainability Working Group has done, they are currently led by Dori Main, but they've had a larger cast of members there. So the sustainability survey that I just mentioned in the previous slide. Next up, they went to essentially to provide a deeper dive into sustainability for a subset of the AgBioData databases. So that first involved identifying possible candidates and then inviting these for further sustainability modeling. Phoenix Bioinformatics produced in the meantime another report on sustainability models for our databases, which is up on our website. And then next up, Dori, I believe, conducted interviews with PIs of representative crop and livestock databases from that identification exercise I described before to identify their resources over time, so personnel funding, challenges, opportunities. So essentially, more detailed interviews. Currently, let's see what Dori Main tells us tomorrow and in her talk, and then there'll be a panel discussion on sustainability in the afternoon, and we will have working sessions Tuesday evening on ways to communicate to our stakeholder groups, essentially. So stay tuned. Okay, so I spent a while now talking about the individual achievements we've made within the RCN grant, challenges that we still have to overcome. But I wanted to step a little bit back and see what we've done in terms of our overall impact for our network. And so one of the ways that we decided to quantify this when we started out with the RCN grant was to hire a professional survey consultant to measure the uptake of fair data or of fair data awareness in our stakeholders. That was one set of surveys, and our databases, another set of surveys. And so what you see here is that we had a 13% increase in our stakeholders in terms of their familiarity with FAIR principles, and a 6% increase for our database professionals. And so you might say, well, that's nice, but we all started hearing about FAIR data at that point. So how do we know it's AgBioData and not just, us hearing this from other sources? And we do think we had some questions to address that. So 95% reported that AgBioData had a direct impact in improving our knowledge of FAIR, and 42% of us implemented changes due to AgBioData. So I think that's actually a really good indication of how we're doing and how we're impacting our community. And most of you hopefully took that survey, maybe not all of you, but you might remember that there are opportunities just to write notes, and this nice one says that AgBioData has provided an incredible and collaborative environment for understanding agricultural databases. So essentially what Lisa Harper back in 2015 wanted us to accomplish back then. One other way to look at our impact of the network, so if you search for AgBioData in the Dimensions portal, which we have access to from the National Ag Library, we see that there's 138 mentions over the past 7 years, even though we've produced just 12 papers on our own. So I think we've had some uptake across the community in the literature. And one other shout out I wanted to do in terms of impact. So I stole this slide from Muskan Kapoor from the single cell RNA-seq working group, but this was really cool, where one of the thoughts that we had when we started this network, or had the grant actually, was that if this could feed on from other working groups and have working groups apply for additional funding for that topic. And so this is what the single cell RNA-seq working group did, and they were awarded a USDA NIFA grant to advance their community. I'm not going to go into detail because we're going to hear about this working group tomorrow, but that's another example of impact to me. All right, so here we are. We're in 2026. We're at our third workshop. And so hopefully I've given you a long enough overview of what we've accomplished and refreshed your memory if you had forgotten about some of these things. And so as David mentioned before, our focus here is really going to be on network and database sustainability, and also on our final push to wrap up our current RCN work. So we're going to hear from the working groups tomorrow and get good suggestions for them, and also think about things like education material dissemination. So now is a good time to leave you with thoughts about what you want the future of AgBioData to look like. What do you want the network to do? Maybe thinking also about how far we've come from 2015 and how things have changed. There was a pandemic right around here. The Nelson memo occurred at this point, where to essentially promoting public access or mandating public access to data sets that are federally funded. And we've had some major upheaval in the funding landscape recently. And so that's changed how we need to think about our databases and our network. And the grant that we'd probably write today would be different than what we wrote before. So with that, thank you for listening to me talk about AgBioData, and I can take any questions. We can start discussing things. Or we could have a break early. Jody, can you do the microphone? Oh, if it's possible.
All right, you were talking about the educational materials, which just last week I went and found, because as I've been curating papers, I've noticed there's I wanted to refresh my memory about what you guys talked about. And I think your slides are great. I think the materials you've made are great. But my issue is if I go to the AgBioData website, I had to go and search under this working groups to find them. There should be a link right on the homepage, educational materials, right? And I think you guys should promote those to like undergraduate, graduate level at the universities. You know, I can try to find a class, I guess, at WSU, those need in, because I think it's critical. And as far as new modules, as a curator, I think one of the things that's significantly lacking is you guys do a great job in describing why you should, the metadata, why you should deposit this and that. But there's a big lack in people's fundamental understanding of how they should write papers, especially when it comes to bioinformatics. They don't cite, they say, oh, we use the reference genome, but never say what the reference genome was or where they got it, or we downloaded it from the reference genome website. I saw that in a paper last week, but never once identified a publication, a URL, nothing, right? We use this tool, but never say what settings. It's just there's a lack of writings on what they should have to write. And then same goes for reviewers as far as what they should be reviewing and making sure when data is deposited. There's a big gap. I mean, I had a class as an undergrad where we just looked at classic genetics papers and we're like, did they provide enough information? Can you reproduce it? Did it make sense? So I think that would be some good components to add, how to write a paper and how to review a paper. And then you brought up, translating that. Have you guys considered changing them to AI voices? Because there are tools now where you make a typed script, use AI voice, and it will translate it to a foreign language for you.
Oh,
So there's tools out there, but my 2 cents. I would be willing to help out.
okay. Go ahead. Yeah. Oh, okay.
Thanks, Monica, for for the nice overview of AgBio and all the work that has been accomplished. One question I had is, has there been an analysis? I'm sure there has been, but an analysis of who are the actual users of the different AgBio databases? Where are they coming from? The number of users? I think that's sort of cumulatively for all the databases. I think that'll be a very useful metric. Are there some databases are super heavily used, and those are, I'm imagining sort of talking about sustainability for tomorrow. If databases potentially, I don't know about AgBio, but if they're moving towards a model that requires some kind of a fee from libraries or institutions or whatever, are there some of these databases that could be considered sort of the bread and butter, and then the other ones can piggyback in terms of providing additional supporting resources and so forth? But anyway, the question is, has there been work done on sort of analyzing the usership, who they are and how many they are and so forth?
Yeah. No, right? Not across databases, right? Mike, Lynn.
To ask the question of how many database people here have surveyed their own communities? How much?
Like who uses Google Analytics basically, or that like the
Yeah.
Yeah. Although I'd have questions for you, Omar, about using Google Analytics, because it's not like it sounds easy, but then at least for us, like filtering out the bot traffic is like whack-a-mole. So right. And then comparing across, that makes it more difficult to do that sort of cross resource comparison if it's not the same site. Sushma, go ahead. Will we be able to hear her? Try it out, Sushma, and we'll see if we can hear you. And if you online can let us know if she's talking, because we can't hear anybody.
So I have a question, Monica. So I think so far we have been in the AgBioData community, mostly database people and data scientists. We need to maybe expand to the community who are data generators and graduate students and people who are writing actually papers. And maybe we can have, maybe with ASVB or something, maybe some of the discussion panels, how effectively to use these resources, and also from the perspective of writing scientific papers. And we can have like editors, we can have senior scientists, we can have publishers, and engage the community in that way. So I think that could be one of the way AgBioData can move forward.
Yeah, no, that's a great idea.
And I'm happy to help on the behalf of Genetic Society of America. So I am associate editor for the computational biology section. I think Tanya is also there, so we can pitch in from that perspective as well.
Yeah, Oh, great idea. Any other thoughts? Your group now? Anything online? Okay. All right. With that, thank you. So we'll take a break until 5:15, right? 5:15. Got it. All right. So online, see you at 5:15 for our next speaker, Marcela Tello-Ruiz, on funding, current grant writing and funding attempts. So thank you. See you then.
First, I'm going to give you a little bit of context, what we need to fund and why. And then I'm gonna go through the comparison of these opportunities, these four: Sloan, CPAUS, CSSI, and NIFA. Okay. The team behind all of these, it's made of David, Nick, Jennifer, Fiona, Adam, and myself. We also have some contributions from Sushma and Sunita at the beginning, and John. Oh, wait. What? There. Okay. Okay. So there are 6 goals, and they represent the core of what AgBioData needs funding to accomplish over the next 3 to 5 years. And some of the points that I want to emphasize about these is none of these goals fit neatly into a single grant mechanism. So we may need to apply to several. The fair data standards and AI, the fair data standards and the AI readiness, for example, might fall within NSF territory. But as I understand it, some other things like sustainable governance and community coordination are more are things that only specific opportunities like the FIROS and NSF or maybe Sloan in the case of governance might fund. And there is the training and capacity building, which would fall can stretch across both. And how did we come up with all these 6 different areas? We did a little survey among the working groups and came up with we asked for their top insights from working of the past years, and as well as their top priorities. What would be their 3 wishes? What would be if they have infinite amount of money? So we came up with very, very interesting ideas from all of them, and combining them, we saw these commonalities and these special things that we could put together for some funding. There were also some other ideas, some that came through our external advisory committee, as well as some of our colleagues, and some of them are very interesting. Oh. No. Yeah. Okay. Yeah. So I don't really want to read over through these because I want to leave more time for discussion, but I'm kind of just introducing them to you. These are the 4, this slide answers the why now question, and which is one of the most important framing for any funders. Dori is going to talk about more about this in her sustainability presentation tomorrow. But from these boxes, we can see that we know that our RCN is ending. It's August. And we have done the diagnosis work. The consortium is ready to implement, but the mechanism that fund the coordination is winding down. So we need either bridge funds, or we need to start looking at longer term. And we need to start looking at longer term. The AI inflection, this is the urgency case for Sloan specifically. especially the large language models and the AI tools being built today will either incorporate agricultural data standards or they won't. And that decision is being made now. So not in the next 5 years. It's AI, the needs are here. There is this biomedical comparison, where, for example, we just have our paper published for our proposing RSIDs as standard identifiers. And this is a concrete example. RSIDs have been used over the past 20 years or so in medical catalogs, GWAS, CLIBAR, LIDBAR, and this is what makes data machine readable. We showed in this paper that we can do this in implementation. And I was very proud that in the polls for Monica, we were the winner, like four different resources were adopting these standards. And then there's the federal gap. This explains how the NSF as well as USDA fund the technical infrastructure, databases, pipelines, software, but not federal mechanisms fund the coordination layer, other than the FIRES that I'm gonna show our work. And this aligned with working with journals, training authors, convening the community around shared norms, where maybe Sloan would be a better fit for this. So what are the options? Let's see what we've done. We started getting this group of brilliant minds together to get behind these our grant writing efforts in January this year, and we really got only a couple months to make the deadline. In February, that was in February. Sorry, we started meeting in February, so we really had a very short timeline. And the FIRES, you can see, it doesn't provide very much money. That's only $200 for a year. That could hardly pay for one salary with fringe benefits and all that. And we had not very many resources available to do this in such a short amount of time. There was no much incentive. So our little experiment didn't work. We didn't get to submit the grant, but we learned a lot during that process. Sorry, there. So we had it planned, we feed all of these, we were a good fit, we were well positioned for it. But again, we had a very short timeline, we didn't have the resources available when we needed them. And this includes, several things that we're very well aware of, like individual projects have their own needs. There is we needed a lead PI behind the effort with a strong track record, and we were asked this FIRES opportunity requires an important software component. And so how to support that, those funds weren't enough for like a full position to support both a coordinator and a software developer. So it had to be like partial funds. It was tricky. It needed a lot of coordination, a lot of people coming together and saying, maybe sharing resources, maybe coming up with like strategic decisions. And we were really short. So unfortunately, we didn't get through, but we learned a lot, and we got this really strong team, and we got ideas and established the framework for collaborative grant writing. Hopefully, that's gonna continue. And these are the four opportunities that are now we're exploring. We have a couple. Give me a second. There. So we have again now this one is the next one I took to myself to get moving, but I'm gonna show you again that I'm stuck a little bit. The nice thing about this is that well, the similar thing about the sloan with the virus is that it is relatively small. So again, we have to be to a focus project, we see that for Sloan and CPAOs, the deadline is in a rolling basis, so we can actually, apply it now. That's another nice thing. For the other two, we have until December, but we better get started now. At least, my position is funded until August. Yeah, so they have different focus. The largest potential one, up to 10 million, it's the most competitive one, and the scope is more constrained to agricultural outcomes. The CSSI seems to be like the right home for sustained infrastructure investment, but it's very competitive, and it requires a mature proposal too. We can go, and these are just the four most the ones that we're the stronger candidates, but there are other ones like NAPPN, and there is also like the matching funds ones, the FAR, and some other that we're also considering. There's the Gates one, but it's only like 100K, and so these are our best. Okay, now my little stock experiment is I didn't want to bother my team with writing this LOI. So this is my learning experience, really. I went and I used a little bit of AI to figure out, like, what would be the best fit. Sloan has all this like technology track program, and there is this other science track, and the science didn't fit any of our goals, what we want to do. So technology seemed to be right, and according to AI, the open science call was the better fit, the area. It turns out not. And this was like, good intelligent humans from my team at Phoenix Bioinformatics telling me, like, I don't see the alignment really. And they were totally right. So this is what the Sloan, this is where our goals are. Oops, sorry. No, there. So I thought this is really good. We have, it's a small funds, but it's a focused project. We have Phoenix has a track, is a nonprofit, so, and we could fund the governance and government settings, so everything looked good. The challenges became really, to identify the right alignment of what they are willing to fund and what we want to do. And so the priority is really to confirm, to identify the correct program. And I'm going to show you the programs now. But before I just show you what I have chosen. What I was working on was like, I have I chose of the 6 goals that I showed you at the beginning. These 2 were included, the fair at publication and the AI readiness. So I chose these 2 because I felt more familiar, more comfortable, and I didn't have to do, I don't have the software development track record behind me to support me. And I felt like, oh, this is easier. I don't have to have a person. But that's a failure on my part, because the program, actually, the call really requires the software development, but yeah. So it's another. It's a good learning experience. The LOI would need to be changed, and I'll show you. It's not, again, using AI a little bit. I read through the things, but it's a little hard to match these, to align. If I don't have that track, that experience, that much experience with writing. And these are the 3 programs that Sloan has. It's the open source, the one I wrongly identified first. There is scientific collaboration, and there is exploratory grant making. And then these are the 3 different areas for each one of them. And so, according to this analysis I did on using some AI tools, now it turns out that it's really weak. The LOI of what I wanted to do is really weak for the 2 of them, and the possible one is the AI and science. That's the most. So now we're thinking that this might not be the strongest to go for. Still, I'm going to explore with the team, but this is the kind of exercises that we're going through, trying to identify how to fund, how to keep AgBioData alive and going strong. So this is again some of the points that I show you in that comparison of the 4 opportunities. The CPAOs in a little more detail, up to 5 million, and again, this is the rolling deadline, the open science cyber infrastructure, and yeah, so some of the challenges that can be diluted, and this is probably our best next step. Then there is the CSSI, the one that is not that much money and focus. Quite it tracks. One thing that we discovered looking at this grant also is that they have like a sustainability track that is very interesting, because it funds efforts to become self-sustainable, to explore those. And so that's very interesting. I'll show you, I think I have another slide just for that. Not this one, this is the AFRI. Sin track, we feed some of the priorities areas, the agriculture, and yeah, so the NOFO has not been released. But yeah, is one of the good ones. And but for this also we might need to convince one of our USDA PIs to be part of it, to make things easier. This is just a comparison across of a fit assessment of what we have. Again, the 6 goals that I showed at the beginning. With the 4, and what we're selecting. Yep. So this is how the 4 opportunities might work together. We have for the community. For the community norms, maybe the Sloan could do. These are the ones that I wanted to propose, but not necessarily that they would fund. They just might fit. That is the NIFA with some of the things that we could include over there, and the cyber infrastructure one. So again, this is additive layers, not competing alternatives. So it may need more, but yeah, just try a few things. So the plan right now might be something like go with something like Sloan for the immediate term, and start preparing for short term, and in parallel the other 2 that are due later in the year. I think that gives us. Yeah, so I think these are the questions that I really we would really want our help with, like your brains, your ideas, how can we get there? What research community needs are we addressing? What's the evidence for that? What will we deliver for the broader community, as well as the specific and impact and the impact it's going to have in member stakeholders. Dory is going to have a session which is going to be target different stakeholder groups. How will we measure that success? Quantifiable metrics. How will it be sustainable beyond the award? And who can take the lead? Who can support this collaborative grant writing effort? Who can be key personnel, the LOS, letters of support. Yeah. Basically it. That's my last slide. Do you have any questions? Yeah, I should have gone into that more detail on that one, sorry.
I just asked to see that slide, Monica here. So, because I think it's important to see what the ideas were about the meat of the grant, right?
Yes, yes, these are the six. So we gather feedback from all of the working groups, current and even the ones that are completed, and with those like top 3 insights and top 3 wishes. What was the word? Yeah. What these were like the 6 core components, what they had in common and what we wanted to, where we see what we can do and what we want to do. We just need some help, some funds for it. Tanya?
Hi, this is Tanya. I was wondering, when you were gathering the ideas, maybe it didn't make it to the top 6, but was there something about shared tools and shared infrastructure support?
Yes, I can show you actually. Oh, I have to, I'll pull it out. Yes, definitely. Yeah, I just showed like 2 examples of these, but like here, I think there is some here, like workshop to build our, yeah, no, not in this one, but yes, there is. Doreen? You can unmute your microphone.
Thank you. So I was curious in these discussions, there is, the things that you've go back to that thing, the top priorities so in the goals here. But did the committee actually say, do we really need? Like, so for instance, Marcela, your position as a coordinator, I think is really critical. So if you ranked what we need to continue as a group and to function, I mean, did the group actually sit down and think about that in context of what are the priorities, right? Because for instance, one of the priorities is right now getting a coordinator funded to at least continue the things that have actually been happening. And are there ways to consider stopgaps or other items for that right now? Because when we're looking at things right now, and then did the group also consider of whether it's not going to be one grant, but maybe multiple smaller grants that are going to need to support the sustainability model?
Yes, that's under consideration. We really only went through the whole evaluation exercise with the virus, and that's the only one that had that research coordination component, which is not central. It has to be. We had to also demonstrate research goals, not just coordination. So it went from RCN to an actual research proposal, but we were prepared to deal with that. So even the program coordinator position, it was assigned to be 50% project coordination, 50% data curator, data analyst, whatever. Except if it was me, it couldn't be a software developer because that's not that's outside my skill set. But we needed software development, and the funds were a little tight, so we had to be strategic about it. We were thinking we could get a consultant, on the side, and we calculated how many hours we could afford of these for specific projects, yeah. So that's the one grant. But if we were also to combine different grants, it's also a matter of how we would work to pay for that the one person, it have been one of the thoughts have also been to have databases partially funding this coordinator position, but that logistically it may be complicated because little bit coming from different institutions to what institution is the employs that individual. It's not straightforward. There is also, we have say, well, maybe for this we definitely don't need 100% a software developer. So it could be a developer that's already employed. We could pay part of their salary, but that has to be incorporated. Their boss has to be a co-PI or maybe the lead PI of one of these grants. So there is a lot of engagement, and that's what we're here for. We want to see who might be in a possibility that it doesn't take so much time for a single person, but combining efforts here and there, we can come up with strategies to make things for. We know what we want. We just need a little bit of coordination and incentivize this effort, because as you saw, AgBioData has done a lot, and it's important. It's becoming much more important now that we are all heading to the IA era, or we are already there, but our data is not so much. Sunita, did that answer your question, Doreen?
Somewhat, yes.
Hello. Yeah, it's working. So yeah, actually, this is really good. And I'm just thinking about it, that these are the 6 goals that we have here. Maybe we can, for the future grant writing, we can have the products from each of these goals, that whatever we have done so far. And then we need to think about it, like how can we have a more robust product from there that should be from community for the community. So something like that, we can say that these are the data types which are ready for AI readiness, and all those like metadata information and everything. And then we are going to develop these tools that will be used by the community for the community. So that way it will be like more consortium type of that project. And it will also show, it will also be like a research component, like, something like that. And yeah, so that is something we need to think about it. So using the products that we have already developed. So that way maybe I'm just thinking about how can we use that for future.
Yeah, definitely. We need to have like things that are going to be useful to all our databases. So there is that incentive that I'm going to invest here, but I'm going to get a return. This is going to be really something I can use to make my databases more easier, more fair, and widely used.
When we are here, maybe we can get some, more like use cases or some of those things that we think that community needs. So that can be useful, for future grant writing, like, from plant breeders, from animal breeders, so from different, from their lines, that what can be used, these are the use cases that we think these are really important. And then we can choose from those use cases. We can refine it further. Yeah.
Is this working? So hi, this is Lenore. So yeah, so I'm sort of tagging a little bit on like what Tanya had sort of talked about. So we talk about the community. So the communities are stakeholders, and what the stakeholders want, of course, is unlimited, easy access to all of the data in all of the world, at their fingertips, without having to learn how to use like 7 different databases. There's also what we need as a community. We are also part of this community. So how can we help make ourselves more sustainable by collaborating and working on some of these common solutions? I mean, I keep going back to like Lisa's paper and to all of these sort of common kinds of threads. And so, there's opportunities. I think it was like James Colts when he was at from the EAC saying there's so many different great grant ideas that are out there, tools like the Texpresso for natural language, AI-assisted biocuration that would help make all of our databases more sustainable, the AI readiness that will make our users love us. And so, yeah, we have to figure out, what the resources are that we have at our disposal. What do we sort of already have, and what can we build on? I mean, I think the model that I think about a lot is with the Alliance for Genome Resources and Paul Sternberg just kind of going like, okay, we're going to build this thing, and then we're going to just make it so that everybody can use it, right?
Have you talked to every program officer? Huh? Oh, hi, I'm Fringy. Have you talked to every program officer for each of these opportunities, and have you found somebody who's been on a panel previously for each of these things? And then my other thought was all of these are US. Are there international programs that you can look at? And I'm thinking of things like APHID base, which has a different kind of funding model in France.
No, we have not looked at the international, and we have only spoken with the program officer for the virus. For the next would be like the ones that we're looking into. Yeah, but we haven't, only the virus one. And the panel, that's a good question there. We have some people that have been in panels here that we're getting some feedback from, but not specifically on these ones. Thank you. I'm going to share that document that for Tanya's question on the shared tools. Yes, there is mention of shared tools in like by 3 different groups, and I don't know, maybe it wasn't that obvious on the site, but maybe I have to include that in there.
I'm David Molik. It's my pleasure to introduce Dr. Amy Klegarth and Dr. Justin Koss, who are from Element Biosciences and are sponsors, part of one of our sponsors for this annual meeting. I'm really excited to hear what they have to say. I'm going to let them take it away. Really cool.
Thanks so much, David. I guess quick introductions on our end. So I'm Amy Klegarth. I'm the territory manager for Element Biosciences in what we've broadly deemed the Northwest. It goes from Kansas up to Alaska, including some provinces in Canada. So if you live somewhere between here and Alaska, I probably am your Element person. Justin is one of our field application scientists. I'll let you.
I'm Justin. So as Amy said, I am a field application scientist. So I am responsible for helping all our VD customers get their applications up and running on our platform, get their old workflows up and running on the VD, help them troubleshoot, work through new workflows, and explore the numerous applications that we have. I'm based in the Midwest, so Kansas is my state as well. I'm based in Lincoln, Nebraska. I don't have quite as a wide region as Amy, just all the way up to Winnipeg and then down to Kansas. So excited to talk with you all today and introduce you to the VD.
Awesome. So to start out, just to kind of set the stage, I know a lot of you have a background in data science and informatics and are thinking about, questions of data handling. And when David first approached me, I had to kind of think about where we fit in all of this and, like what the story is, what the message is. And so what we're going to talk about today is more on the upstream side of what eventually becomes the data and the data storage and handling. And that's the generation of your genomic data with the technologies that Element has, and that I think in many respects fits into some of the questions that you're all thinking about and how to kind of future-proof, have different efficiencies put in place, and have some kind of standards that go across, groups within your discipline. And so hopefully, by the end of this, you'll know more about the technology and what's available kind of upstream, and then how that relates to what you're doing downstream ultimately. And so I'm going to start us out with, let's see, how do I get this? Is it this guy? Okay. Okay, great. Thank you. So to kind of set the stage, when we're thinking about using things like genomic selection in the ag space, are we really maximizing all the opportunities that are out there? Right now, many breeding operations are still utilizing microarrays, and there's some inherent limitations with that. Some of what you will see is, fragmented data from different disparate methods, slower workflows, slow and expensive assay redesign that comes with those, as well as limited potential discovery, right? You're limited to what you have known and designed around. Thank you. What? There we go. Is it stopping advancing now? It shouldn't advance. Okay, great, awesome. And running into challenges with questions around complex traits and the like. And when we think about how can we solve some of these problems and where can groups like Element come in, some of that comes into making different data types more accessible. And in our case, we're talking about maybe genotype by sequencing. So traditionally, genotype by sequencing has been, prohibitively costly. That's why most groups are using microarrays, because they have gotten enough of the answers at a price point that is palatable. But as we've seen, sequencing costs, data storage, and the like have continued to come down, and it's allowed us to look at larger data sets and larger data types, and running things like low-pass genotyping or genotyping by sequencing and looking at imputation are now more and more accessible. One of the other ways that we are working in this space is to automate things like Hive capture workflows that are typically kind of burdensome on the wet lab side of things. We do this with something called Trinity that Justin will get into a little bit more. And just offering different flexibility in terms of how you run your NGS solutions. And I'll show you how that's kind of played out across our platforms. And finally, thinking about access to more local and decentralized NGS resources. So the industry and these different spaces continue to ebb and flow. We go to big, bad industrial sequencers that everybody sends into, and then we see challenges associated with that. What I think Element has created is something that is flexible on both the low and the higher ends and lets you kind of adapt to your own needs and see kind of replication across sites if you want to, or a way to build out within your site if you need to. And so the platform that I'm going to talk about the most today is the AVD24. We also have a sister platform to this called the AVD. That's an NGS only platform. It has slightly lower output, just kind of setting the stage. In many respects, they're very interchangeable on the NGS side. And some features that I'm going to get into in terms of just some of the background of the technology, the underlying chemistry that facilitates it. we'll ultimately get into how we can make these workflows more affordable, increase accuracy, increase the data quality, increase flexibility, and just give you more control over how you're utilizing this type of platform in your different workflows. To start out with, I just want to kind of set the stage and context for where we sit in the NGS space. And so a VD24 comes in at a really reasonable and lower cost per gigabase in terms of your data generation relative to the other low and mid throughput sequencers. You really have to be at that really high end NovaSeq X, X Plus to be getting kind of a better price point. And what that means is you don't have to invest in $1 million platform to be generating affordable data in your own hands or across labs. And we're pushing this even further with a platform I'll speak a little bit more about at the end called Vitari. And so it just really has been a main goal of the company to really try to democratize sequencing and bring sequencing back into the hands of labs and organizations that have felt like they had to be sending it out for the longest time. And when we talk about flexibility on the platform, this is just the basics of how both the AVITI and the VD24 operate. So the platform has two sides. Each of these sides is totally independent. So that really offers the ability for different projects to be running at the same time. When one side is started, doesn't dictate at all when you can start or stop the other side. So you can be doing two totally different things on the platform at the same time, or just using it to boost your throughput for something that you have higher throughput needs for. We have a variety of run formats that are standardly seen across NGS. I can get into a little more about that later. But then we also have individually addressable lanes. So within a flow cell, you have the ability on our 2x75 and our 2x150 formats to either replicate your indices across lanes A and B, or to run different but similar enough projects at the same time if you want to split your output. And what I'd like to get into now is a bit about the chemistry and how this has been something that has really allowed Element to innovate with and really reach things like price point and quality that hadn't changed in quite a long time in the NGS space. And the start of that is that we use something called rolling circle amplification. So we use rolling circle amplification. This is in contrast to standard NGS, which uses sequencing by synthesis or bridge amplification. And so we're not using PCR here. And because we're not using PCR, we're only ever making copies of the original template. And so we can still have an error propagated, but it's not going to get propagated exponentially as it would in a PCR-based workflow, right? So your one error that might be generated at a time isn't likely to be perpetuated throughout the whole polynear cluster. And so because of this, we don't have on-instrument PCR error. We have reduced the optical duplicates, and index hopping is really negligible. Ultimately, we generate about 1,000 copies of each template, and these get clustered down into what we call a polyny, or you might be familiar with the term cluster if you're thinking about Illumina or other sequencing by synthesis. The second part of the chemistry is that we use something called avidites. And so we call this avidite-based chemistry or ABC chemistry. And these avidites are a dye-labeled polymer that you're seeing on the lower right. And they have what you can think of as these arms. And these arms are coming off and they're binding DNA start sites. And then the label on that polymer is what we're ultimately reading with our optical system. So each avidite corresponds to the nucleotides that we're reading. So A, G, C, and T. And the multiple arms are a key factor in some of our accuracy and our ability to reduce costs. And that's because if only one of those arms is attached, it's maybe attached to one of those random errors, it's going to wash off. It's not a tight enough construct. But when you have multiple arms bound, it's going to form a very strong bond, and it's not going to wash away, and that's going to get read. And so there's some self-proofreading that's happening within this chemistry. And ultimately, that's helping to reduce our error rate and bring us to what is a Q40 accuracy. And this is, a pretty in-depth contrast between SBS sequencing as well as ABC sequencing. But what I'd like to kind of hone in on is with that avidite chemistry, we've been able to reduce the reagent consumption about 1,000-fold. And that's what helps us bring the actual cost down, because we don't really need to use nearly as much reagent to accomplish the goal of sequencing in the long run. And then the other thing, which I'll call out a little later more thoroughly, is that it's compatible with 3D sequencing. So it allows us to explore a third dimension, or these Z dimensions, that enable us to get into both single cell and spatial genomics. And so it's just a more adaptable type of chemistry that allows us to get into more multi-omic applications that SBS is kind of more inherently limited by. So these are just showing some data on both optical duplication rates and index hopping, because I don't want to just tell you that we have low optical duplication rates and low index hopping. Optical duplicates happen when a large cluster is called as two separate clusters, or from local reclustering once a molecule is released from polymerase copying. And what you can see is across element libraries, we have really just negligible optical duplication rates. Similarly, when we look at index hopping can be a huge issue in terms of data integrity. This is happening when you have indices that are misassigned from sample to sample, and it becomes more problematic on systems that utilize pattern flow cells. So these might be things like the HiSeq, NovaSeq, and the rates of index hopping typically are anywhere from 0.3% to 3%. So it can be, a non-negligible slice of your data that's being impacted by it. And because of that avidite-based chemistry, as well as that RCA component, we're seeing really minimal index hopping across the board. In general, we are achieving an average of Q43, and importantly, we call these on individual bases. So a lot of technology has actually been their quality scores. And so there's ambiguity in terms of what those algorithms are and how much of the data actually falls into that category of Q30, Q40, et cetera. Since we attribute our quality scores on a base-by-base level, we're a true Q40. So our error rate is about 1 in 10,000 bases. And we also have chemistries for PCR-free whole genome libraries that get into Q50 plus. And so if you're really having an application that has high sensitivity needs, that you really need to dampen the background noise, that's another kind of gas pedal that you can push with our platform and with our chemistry. This is showing how the accuracy level, the Q40 data, translates into higher accuracy across different coverage levels. And so what you see is at the lower coverage levels, the differences are exacerbated. So when we're thinking about the ag space, where you're pushing lower and lower coverages and utility and things like that, you're going to see kind of more bang for your buck from that higher quality data at those lower coverage levels. It's going to make a bigger difference at those levels than the deeper coverage that you have. With that, I'd also like to call out the performance that we have across some difficult regions of the genome, which are especially low and especially high GC content regions. Again, in the ag space, we're often seeing folks exploring things in the microbial realm, microbiome, things like contamination. And a lot of the microbial workflows often have organisms that have been characterized with higher and lower GC content. And so this is an area that, again, that RCA, that rolling circle amplification, avidite-based chemistry is super robust through. Similarly, it's important to call out in those types of applications where we have longer reads that we're using, 2x300 in this case. Traditionally, you see a dive well below the Q30 threshold in read 2. We also have a quality dive that happens in read 2, but because we're starting at such a high threshold, we're starting at Q43. What that means is we actually never go below Q30 in our quality across those. So you can read a fuller length sequence with reliability with this chemistry. And similarly, when you look at variant calling accuracy, we see over a 50% reduction in false positive, false negative rates between SBS and our chemistry. Another area to distinguish these data types from kind of traditional sequencing is that the avidite-based chemistry and rolling circle amplification allows us to utilize larger insert sizes, so we can go to 1 kb and even above. And that ends up having downstream ramifications for assembly and assembly accuracy and statistics. And so what we see is a large reduction in errors in these assemblies, many of them falling into that category of false negatives. And that's because bioinformatically, even if you're still sequencing 2x300 across two different sequence types, 2x300 with a larger insert, so a larger gap in between it, is bioinformatically more meaningful and more complex than a smaller insert. And so that's where you're seeing the improved assembly statistics across organisms and workflows. Similarly, you're seeing Q40 adding, reducing costs in that it maps more efficiently. And you'll also see this played out in a variety of metrics. This is across both DNA and RNA workflows. And so you see the increased quality score, lower duplication rate, higher aligned read rates, and increased mapping quality on the DNA side. And on the RNA side, you're still seeing that increased quality score, the reduced duplication rate, and comparable in the aligned reads. And that 5' 3' bias that you're seeing is because of that ability to read through the high GC content. So the 5' end tends to have more concentrated GC content in mRNAs. And so that actually shifts that statistic for that. And so that really just kind of becomes a proof point for the ability to read through those GC regions. In terms of workflow overall too, I think one thing Element has done a really great job of is making the technology so that it fits with existing workflows, both before and after our portion, which is the sequencing primarily. And we have something called freestyle chemistry. And that means that if you have a P5, P7 library, or your collaborators have that in their freezer, that you're going to be able to take that out and use our freestyle chemistry and go ahead and sequence it again. So you also don't have to change many of your library prep types. If it's a standard P5, P7 library, it should sequence just fine on this freestyle chemistry. And then at the end of the workflow, we're not spitting out any kind of special proprietary data type. We're exporting a basis file. You're going to run basis to FASTQ and get a standard FASTQ and fit into all the standard pipelines, right? We're really not trying to force your hand into anything kind of proprietary in that sense. This is just an overview of, some samples of different application types and workflows and how many samples you could fit between the AVD and the VD24. Happy to pull that up later and show folks some more about that. And then digging into kind of the different formats that we have to run, our 2x75 and 2x150 have the individually addressable lane option, and our 2x300 has the option to be run on the VD24 with something we call expert mode. Expert mode is going to allow you to get 20% more data that's of a Q30 and above standard, so it's kind of bonus Q30 data in that sense. And that's what you would get. That's Q40 that's shown on the slide. And then there's the potential for that 20% or more bonus. And before I hand it over to Justin to talk about some applications and before we get into some of kind of the alignment that we see with regard to FAIR and the questions that you guys are addressing at this meeting, the last thing I'd like to call out is the multi-omics capabilities that a VD24 offers. And so on the multi-omics side, we're able to interrogate DNA, RNA, and protein, including phosphorylation state. Right now, we're working specifically with cell-based assays, but later this year, we're going to have tissue enablement, so FFP and fresh frozen. To start out in the ag space, we're going to be most relevant to mammals, so various livestock. There is roadmap for doing some custom morphology markers, and that's kind of the inherent catch. Our morphology markers are designed for mammals primarily. You should be able to swap out some custom markers and enable that for other organisms, birds, and the like. Plants, I would say, are probably further down the roadmap. There's just some more challenges getting the chemistry past the cell wall and different fluorescents that can come up. So I think, they'll continue to explore that, but think about the multi-omic side primarily on the mammalian livestock end of things. But overall, we're able to also do sequencing directly inside the cells. So if that's the direction that any of the collaborators are taking in terms of characterizing organisms, that's something that we can definitely talk about. And so with that, I'll hand it over to Justin to talk about some applications.
Yeah, so we'll dive into some applications now. So you have a sequencer. How do you use it? What applications can you run on it? How can it make your life in the lab easier? How can it make submitting data that's fair, appropriate, easier? So we'll dive into really the agricultural genotyping problem. And I spent a lot of time in this field prior to joining Element. I worked designing products for genotyping and did a lot of genotyping during my PhD. And in ag, there's a significant problem that we all experience. One of them is multiple platforms and there's zero standardization. So working on the array side, from my background, there's a GGP 50K, a bovine HD, there's genotyping by sequencing, there's different amplicon panels from different vendors, each with its own file format, different allele coding, coordinate system, and its own separate software that needs to be installed on your HPC in order to be used. Additionally, data is often trapped behind these vendor platforms. You have to subscribe to them in order to get access. They have their own encodings. And then those fixed marker sets like arrays don't evolve over time as the genome reference expands. So a lot of researchers are developing new methods, publishing their research, a whole new genome assembly, but that $1,000 experiment was purchased on an array from 2 years ago, and you can't go back and re-impute to the latest reference because you don't have base calls or FASTQ files that you can generate. Then there's also the reproducibility gap. So a lot of these array platforms, they have these black box analysis pipelines and proprietary algorithms that don't let you be in control of your own data. So that's why we really come in, and Element has a really unique system, and NGS as a whole, where a lot of the field is moving to NGS for genotyping. And we have some really key points now and new levers with the technology that make it a really good use case to move to NGS for genotyping. First, we're dealing with a richer data set. So with genotyping by sequencing, you're directly sequencing 120 base pairs of fragment of your template DNA, not a linked marker, not anything nearby. It's your direct sample. It's adaptable. So a lot of genotyping by sequencing, it's panel-based approaches now. So you have DNA probes that go in and bind your genes of interest. So if you've heard of exome panels before, a lot of vendors are now having custom exome panels for your species of interest, your genes of interest. A lot of universities have their own capture panels of interest for cores to rinse and repeat their research, their genotyping platforms for their organisms. Additionally, with genotyping by sequencing, there's less bias. So it helps reduce that ascertainment bias that we know exists in arrays. And it's also future-proof because we can go back and re-impute to those whole genome reference assemblies from our FASTQ data. You may be asking, how does sequencing data compare to array data? Is it orthogonal? Is it concordant to array data? And the answer is yes, it is very highly concordant. So NGS is able to recapitulate microarray calls with 99.5% concordance. Shown on this plot is an example of I think about a 4 megabase region. Oh, sorry, this is 1.6 megabase region in the bovine genome. And this is from a target capture experiment. So is everyone here familiar with target capture or hybrid capture also sometimes? So you have a streptavidin oligo that binds your DNA of interest, you pull it down, you're only sequencing your library of interest. So we have regions that are specifically targeted. It's not a whole genome shotgun application. Oops. All right. So what we're seeing on here is that we're getting great concordance. The blue bar on the top, how do you do this one? Oh, is it the star? Okay, yes. Okay, so what we're seeing right here is your NGS sequencing data. So this is aligned to your reference genome, And shown below are your microarray calls. And we're seeing great concordance across duplicate arrays. 2 and 3 are 2 different array batches. And number 1 is your NGS sequencing. So this is from a low-pass experiment, or sorry, target capture experiment, great concordance. Now, what happens when we get into sites that aren't concordant? What are we seeing? So these sites are really interesting. What about non-concordant sites? So when we look at our twist capture, so shown at the top here, and our 100K array that's aligned to it, we see some gaps in our assembly here. So we see some regions that aren't concordant. And what about those gaps? What do we say about those? We have 2 discordant calls here. Is that a het or a homozygote? We can't compare those back because it's a fixed marker array. So from the array, when we're comparing the array to our sequencing data, we can see that the array missed 4 calls, and 3 of those were actually hets. So if we were using this array data in our breeding program and going back to this in the future, we'd be introducing errors into that downstream analysis because we weren't working with the actual truth of our sample. So again, right up here, we're seeing 5 out of 31 heterozygous calls in that 4 megabase region. So that is showing a significant concordance issue, 16%. So that leads to 3 no calls and 2 discordant calls. So things that we don't want getting into our data set. We'll dive in a little bit here. So this is discordant SNP 1. So arrays can make the wrong call when the adjacent SNPs contribute to that signal variability. So we all know about those linked markers, those linked SNPs that tail in the region that can have an impact on our sequencing data. Here's an example where we can see that from our data. So the position is heterozygous with 29 supporting sequencing reads and 3 adjacent supporting SNPs. So we are very confident in that sequencing data that we're using for that het call. So not something that we'd be able to capture on a microarray, but because we're moving to sequencing, we are able to have that on there. To our next slide. All right, so now we'll move into this example here. So here's an example where it's really asking, is this an array manifest error or is this a problem with our array signal? So here we're seeing that SNP position at that A in the reference genome and our twist probe sitting on there. And we can see that from right here, the sequenced variant is 1 base off the array coordinate. You can see how the array had that. Right here is the array call, but right here we're seeing direct concordance. So that adjacent SNP could be impacted by the probe. So one thing that I do want to note here is that if you were designing, say, a new probe for a microarray based on this data, and you were sitting down, you didn't have this information knowing that that was a het, and you designed your probe assuming that it was a homozygote, you would not capture that het, and that would be another variant that was not entering your pipeline. So one thing that we have that really revolutionizes the target capture assay is Trinity. So AVITI, I said, and Amy mentioned, has a lot of tricks up its sleeve. The target capture approach that we just shared, where you're getting discordant array calls backed by sequencing data and using newer methods to investigate those calls, we can use with our workflow called Trinity. So Trinity enables on-device hybrid capture, reducing that bench portion from up to 16 hours of hands-on time into as little as 1 hour. So you can go from your bench workflow where you have your heated post-hybe washes, post-hybe PCR, QC, which can take up to 5 hours at the bench and add an extra day to your data time. You can streamline and condense that down into as little as 1 hour and go directly into sequencing with Trinity. There's no sacrifice to quality as well, and you can unlock more sample processing capacity because you are able to utilize that lab time more efficiently from not having to perform the hybrid capture workflow at the bench and letting the AVITI handle it. From a probe compatibility standpoint, so one thing when you are working with hybrid capture probes is to pay attention to the size of the probe panel. So not all probe panel sizes are compatible. We do have a limit of 0.8 megabases. It's just from a steric standpoint. So we need to make sure that you have enough mass on your sample to actually pull down on the flow cell. So we need to make sure that your panel is appropriate to pull that down. But now we'll jump into a little bit of the more exciting meat, which I want to share with is how the AVITI supports FAIR standards for breeding programs. So how does it support your data being findable? So how can you get the data that you generated last breeding season with the AVITI? Say you've moved to the AVITI, you have all your NGS and genotyping data on it. How is it more findable? How is it more accessible? So how can you access your own data without a vendor subscription? Interoperable. So can I manage data between 2 different genotyping platforms? And then reusable. Can I reanalyze last year's whole genome sequencing data against the new reference genome that's been published? So starting with findable. So what do breeders really need when looking at their records and working with their data from sequencing? One, it's structured, machine-readable metadata that travels with the actual data. So any scientist should be able to find, understand, and catalog your data sets years after they were generated. I have horror memories of trying to find old sequencing data, FASTQ files, submit them to the SRA, search through all my old files from the core. It can be a real nuance. What you get today, so you do have that vendor lock-in data. So you have all your raw data that's produced by those IDATA intensity files, where you may need Genome Studio to review those. Not great. Run parameters may be scattered across the lab. Was this a 2x150? Was this a 2x75? Was this on Illumina? Was this on Aviti? Which flow cell size was this? Lot information can be very hard. And then there's no standardized output structure. So we know that every lab organizes genotyping results differently, and that finding a data set from lab A can be immensely challenging, and it can be very easy from finding it from lab B. So how can the Aviti help standardize this? So interestingly, the Aviti has some really great built-in solutions that help make data FAIR appropriate. So every run produces structured machine-readable metadata off the bat. So we have a run manifest JSON, a run stats JSON, metrics, and index CSV that's generated automatically every time for every sequencing run. What's in these metrics is all the run output shown on the right-hand side. So from an Aviti run, you get this structured JSON file showing you your instrument settings, some aggregate QC information that's contained in this runstats.json. You get a metrics.csv file. You get your demuck stats, so you know how many barcodes were assigned to each sample. And then from a sample level, you get all of your project tier information, so you can set up your sequencing runs according to their project. So you have another metadata piece that you can go back to and link for your records. And then you have all your sample information. And the nice thing about this is that this is generated for every run. Every run has the same structure, so you do have that provenance to go back to. Also with the Aviti, you have automated QC reports in both HTML and multi-QC format. So pushing data sets through FastQC can sometimes be a challenge for people. With our basis to FastQ software that takes our base call files and converts them to FastQs, you get an output of a HTML multi-QC report automatically. Nothing to push through FastQC. It ends up in your output directly automatically. And then you get the benefit of having that consistent, predictable output structure with every run. I'll pass it back to Amy, and she'll talk a little bit about the accessibility challenge.
Yeah, so in terms of the accessibility challenge, breeders need ultimately persistent access to your own data through open protocols, regardless of what platform you've generated on. And so some of the challenges here today is when you have vendor hosted results that are through different subscription services on the vendor's cloud, where there's the potential to lose that access to the data with a subscription. Raw data is often unavailable. There can be black box components to this, whether it be the algorithm, you're getting the output after the kind of black boxed algorithm. And so you get the SNP calls, but you're not really sure where they came from. And reprocessing isn't possible. And then platform or chip discontinuation. So as chips change and discontinue, how does that, where does that leave you in terms of continuity of a project or translating over to the next phase of your work? And with Aviti, we're talking again about sequencing and genotyping by sequencing. We have the ability to send your data where you want it to. So you can send that to hard storage. You can send it to your own cloud solution. We do offer a subscription-based cloud solution, but it's just an AWS wrapper, and it's a totally optional, thing if you don't already have your own compute set up, and you can always port it over to something else. It's not proprietary in any way. The instrument is going to send you your base calls, and you're going to do basis to FastQ to whether it's that S3 bucket or other storage. You own the raw data, so you're getting those base calls, you're getting the FastQ and the VCF files. You can reprocess it at any time. So if a new reference comes out or a new analysis, comes into vogue, you're able to go back to that original data or add to data sets and reprocess. And again, there's no required proprietary subscription. We have a solution if you don't have one, but it's completely open in terms of your ability to port in and out of it. And so, it ends up just being more flexible in that regard. Justin?
So interoperable, the challenge. So breeders need persistent access to your own data through open protocols, regardless of which vendor or platform it came from. So today, again, you have those vendor-hosted results, black boxes. A lot of those are subscription-based, and once you cancel your subscription, you lose access to that data. The raw data is often unavailable, so you often get SNP calls, but not the underlying intensity data. So you don't know if that was a quality call or a low quality call. Oh, I'm sorry, that was the wrong slide. This is very jarring. Apologies, everyone. I had the wrong slide pulled up. Okay, so interoperable, the challenge. So standard data formats and shared vocabulary. So data sets from different platforms, labs, and years can be combined. So today you're getting those 15 plus array formats in cattle alone. So 3 companies, 15 plus commercial arrays in under 10 years. Again, that Illumina top bottom format, Affymetrix, AB, plus minus, it's very confusing, not ideal. Managing and merging those data sets can be extremely complicated and require bespoke pipelines. So one of our collaborators, Curio, that I showed the discordant genotypes from with Twist, they have a solution where you can actually have array data and sequencing data combined in their software, and it's able to help merge these data sets together. So if you have a collection of array data and you're looking at migrating to sequencing data, their platform is great for that as well. And again, today you also are locked to those commercial chips with fixed markers. It's hard for you to add custom content for your species of interest. So how does the AVD help this? Again, the AVD one format pipeline. So it goes directly from FASTQ, BAM, VCF, downstream for your downstream pipelines. Of course, noting that from the AVD, you'll have your base calls translated into FASTQs. The AVD does support open Nextflow pipelines on GitHub. So through our AgBio Cloud, you're directly able to connect third-party pipelines into our AgBio Cloud solution. So you can actually stream your data directly from the AVD to S3, Google Cloud Storage. Very, very efficient there.
And R, so reusable. So you need data that gets more valuable over time rather than less valuable. So you want to be able to reuse this data against new references, new trait models, and new analytical methods, right? That is changing all the time, and having data that doesn't move forward with you can really be a challenge. And so array data, in a sense, is frozen in time. When a new QTL comes out for heat tolerance or any other trait, you may not be seeing that. It may not have been chosen. You're exploring the known knowns with these panels, where these are areas that you've preordained, and you're not getting to see anything interesting that might be on the edges. Reference updates can make these challenging to reinterpret and to look at in the future, and you want to be able to leverage new updated reference genomes. And again, those black box analyses can be a real challenge. And so AVD, we like to think of as generating data that can be an appreciating asset rather than a depreciating one. You're going to have those FASTQ files to reanalyze into the future. It's a very comprehensive data type, and it's a very high quality data type with that Q40 kind of standard there. So you have the ability to realign these existing FASTQs against updated references, discover new markers without needing to resequence, and have open source pipelines. Again, we're not creating any type of proprietary data file. You can go into your own existing pipelines and other open source pipelines and enter this data into it. And really just explore a broader range of traits and SNPs. When you think about low pass and being combined with imputation with groups, whether it's using Curio or your own pipelines, you're able to continually revisit these samples in the future with updated kind of lenses. And
and I'll do my best to keep the clicker and the slides in sync. Apologies. All right, so applications, it's this DNA sequencer, but as we said, it has a wide variety of applications. So from wheat genomics, beef cattle, sorghum breeding, whatever you're genotyping, the AVD is here to support it. So one thing that I do want to share is low-pass sequencing. So genome-wide genotypes at scale. It's a very powerful application, and one thing that I do want to end on here. So we've talked a little bit about moving from arrays to sequencing, and this is an example of how you could actually enable that with the AVD. So low-pass sequencing, what is it? Well, it's low-pass sequencing across the entire genome. So some people call this a shotgun sequencing. It's rather than a pre-selected panel. So a lot of times this is at half X, maybe 0.1X. Some folks will use 1X. It really depends on your breed. It can lead to accelerated genetic gain because you're getting more markers across the whole genome in that VCF file. And what this allows you to do is scale and sequence more samples cost effectively. So you could imagine having 96 samples on one flow cell, but maybe if you drop your coverage a little bit, you could scale that to 384 and not lose any imputation confidence on your reference population. So when comparing arrays to sequencing, there's a hidden cost to array-only approaches that I do want to highlight. So one of that is missing variant types. So if you are focusing strictly on arrays to study your biology, you're really only focusing on those pre-selected SNPs that are put on the panel, and those pre-selected SNPs often miss those really complex CNVs, indels, STRs, and structural variants that matter to your genomics. With low-pass whole genome sequencing, you have unbiased detection of all of the variant types that you're interested in. Additionally, with arrays, you have there's a real challenge with population specificity. So in my background, I worked on a lot of different cattle breeds. And when you're dealing with arrays in a wide variety of cattle populations like Angus, Hereford, Holstein, they don't behave the same on array. They have different background genetics, different reference genomes. So arrays are designed for most of them are designed for a single breed or specific unique populations. And they have poor performance often in crossbreeds and some of those composites because they just don't have the haplotype information in them. Low-pass whole genome sequencing, on the other hand, doesn't have that same ascertainment bias, and it can discover population-specific and rare variants in any sample. So again, going back and re-imputing to your reference population, you could identify new rare variants, say if you had sequenced something and then 2 years later, a new reference was generated and it identified a rare variant in there. Again, arrays have the lack, or I guess the unfortunate ability of not being able to be upgraded within 6 to 18 months. So they do have a long development time, whereas with sequencing, you can sequence it once, analyze in the future, or if you wanted to look at some new traits, you could quickly generate a library and throw it on your AVD. So a quick high-level comparison about a fair comparison between sequencing and SNP arrays. From a findable standpoint, sequencing has that standard FASTQ, BAM, VCF format with that rich run metadata and those JSON files that you can use for your fair approach. The AVD also allows and supports the accessibility standpoint that we saw by able to stream your data to your storage of your location. The interoperability that we saw, we do have that industry standard output format and the compatibility with Nextflow pipelines. And then from a reusability standpoint, you can go back and resequence, you can reanalyze and reimpute your data. So again, the main point that we want to drive home is that sequencing data becomes more valuable over time. Like Amy said, it appreciates over time, whereas your array data is fixed and frozen in space. To quickly show you a real-life example, a case study of using low-pass sequencing instead of arrays in cattle is shown right here. So this is an example where we had 100 animals, so 50 Holstein and 50 Angus animals, and we had 7 different input sets, each imputed. So we started with an array, so that was a bovine SNP 50, so 50K array, and then we had a low-pass, or a few low-pass whole genome sequencing data sets at different coverages. We used 2 different imputation pipelines, one for the array and the other for the low-pass whole genome sequencing data. And the metric that we paid attention to was the non-reference concordance, or the NRC. And that asks how accurately those imputed variants match the high coverage gold standard. And shown up here is what we're actually tracking, that non-reference concordance across our different data sets. So we want to have a high concordance to our gold standard. That's the goal for our genotyping. And what we can see is that with half-X sequencing, we get around 0.913 concordance, so mean non-reference concordance to our gold standard. So that would be the nice sweet spot. But if we look at the array, we're seeing a concordance of 0.6, so much lower concordance compared to the sequencing data. And one thing that I do want to highlight is that with sequencing, you can see that as you sequence more, your gains start to plateau a little bit. So this is meant to highlight that you can do effective genotyping at this low pass sequencing level, with 0.5x being a nice sweet spot, depending on the reference genome that you are imputing to. So that is one thing to keep in mind of the reference genome that was used for imputation. These had haplotypes to support the animals being investigated, which helped us achieve this concordance here. So again, at the 0.1, we're already winning. 0.5x is the true sweet spot for this application. And we do retain those low minor allele frequencies where it matters, so we're not losing the power of our data set. And the reference panel that we used is fair ready. So all of these animals can be reanalyzed in the future when a new reference comes available. And any new data that's generated following this method would retain the same run stats parameters, JSON, all the sample provenance information from this experiment. So we would be able to longitudinally study this same question in the future, maintaining our reference our pipelines and our organization of the project. So ending on just sort of high level, the solutions that we enable for these hybrid capture workflows. We do have a variety of partners that enable these Trinity or target capture workflows. So we do have collaborations with Twist and IDT to enable the probes. And then from the Aviti standpoint, we do have flexibility for hybrid capture solutions on the 2x150 or 2x75. The benefit of some of the 2x75 sequencing workflows, so if you are performing genotyping on rare samples, maybe you need next day, 2-day turnaround time, with the Aviti and with Trinity, you would be able to build a genotyping pipeline that could output data that's FAIR compliant in as little as 2 days. So you could go from sample to output data in 2 days, pushing directly through your pipeline. And again, all of this data can be directly streamed from your Aviti to your provider of interest. So you can stream your data directly into your server, so your compute server at your university, your Google Cloud storage, your Amazon S3. We even have our own solutions if you're looking to get something set up and you need assistance in setting up an analysis solution. We have that as well that we're able to provide. I'll pass it back to Amy to end a little bit on our fleet of Avitis.
And the end is near, I promise. We're in the last 3. We're in the home run. So this is just an overview of all the platforms that Element currently supports. And the one that I'd be remiss if I didn't mention at the very end here is the one that's coming out later this year. This is Vitari. This is still a benchtop system, similar in size to Aviti, but it is the first high throughput benchtop system that's coming on the market. And the key things, especially when we think about the ag space where price can be really, really sensitive in terms of the number of samples that a lot of these workflows are trying to get through. Vitari really comes in at a super, super aggressive price point at $190 per gigabase, so $1.90 per gigabase. And at volume, we're looking at $1 per gigabase. So that's about $100 human genome. And what we're doing is 5 billion reads per side. So just like the Aviti, it has 2 sides. We're offering 2x75, 2x150, 5 billion read outputs to start, with some future roadmap to both lower and higher throughput, as well as future roadmap to layering cited profiling onto it. And then the ability to split that 5 billion reads into up to 6 lanes. And so you can really kind of mix and match your workflows to get what you need out of it at a really cost-effective rate. So just be aware that that's on the horizon, it's coming out, and it's, driving down those run costs and the accessibility for genotyping by sequencing even further, and also has compatibility with Trinity. So you can do those same automated high capture workflows if that's a direction you're interested in moving into. And with that, we're happy to take questions. We'll also be around tomorrow afternoon, happy to talk to folks at the break, and thanks for having us. Yeah. Yeah.
in your slides. Oh, sorry. Hi, this is Lenore. I did have a question. This maybe is more to your marketing people, but I'm wondering, like in the scheme of things, when you look at what the consumer, what the user ultimately values from the platform, like how much does the fairness actually fit in, along with the sort of, the quality of the reads, the cost effectiveness? Like, do you ask them, like, does it really matter to you that it's fair?
So I think I would be surprised if it comes up in those specific terms, but I think in the way that the company has wanted to kind of democratize sequencing across ways, it just inherently matches a lot of that, right? They've made it a very conscientious effort to, again, not pigeonhole you in either the front or the back end and try to make things, we're not trying to force you into a box anyway. And I think that inherently kind of fits and matches, which is, I think, like David had been familiar with the platform through the USDA in Kansas. And I think that's where kind of the thoughts came around to this talk, because it is just kind of, we're very agnostic in many ways. We play well with others.
For scientists, and they developed the ability with that.
Right.
Stupid adapters.
Yep.
Like each time we have.
It was in our instrument fleet too. Or the upgradable to the AV. Out on a DVD. How can grow and be upgraded. A lot of flexibility and
I'm Tae-Kwan Ko from Michigan State University. So the past years, 80% of our sequencing sample from ABD. So I can feel how it's revolutionized price-wise and really make affordable. But one small issue that I noticed is that uneven distribution of lead numbers across sample multiplex. For example, let's say 4 sample multiplex. One sample will have 30 million reads, second probably 20 million, third probably 35. It's very variable. And most of the time it's fine because genomics core will resequence them anyway. But when I have single cell genomics data, it's problematic because generating libraries very expensive, and even the yield is very, very small, so we cannot resequence. I wonder you guys are aware of this.
Yeah, so I would say that that's normally a question of some of the whether the QC approaches and how the pooling is happening up front. So I think that's something that maybe Justin can speak to a little more as the FAS. But it's not something I'm aware of that is like inherent to the platform. So I would suspect that with different QC troubleshooting, you should be able to get.
Yeah, from what you're describing, it sounds like a lot of it may be based on the pooling of how your library was pooled together, which may happen at the core facility itself. Typically, equimolar pooling can be used, so you have equal reads across all samples. Some runs, that may not be how the pooling was set up. One thing with the VD that we can do, and that we advise all our customers to send us in advance if they have questions about the run, is a fragment trace. So you could check with the core where you had your library sequenced, ensure that no adapter dimers were present in your library before sequencing. I always advise all of my customers to make sure you do a quick bead cleanup before sequencing to remove those adapter dimers, because that is sequencing real estate that will get sequenced if your library or your sample does contain those when it is submitted to the core. Thank you.
I don't even know if anybody's still on Zoom. Anyway. Oh, there are. Okay, this is Tanya. So I love that you're using open data formats and common data formats, and that you're making it easy for people to go from one to the next and to upgrade. And I was thinking about you saying, okay, we have like all this information about how the run was done, and there are the CSV files and the FASTQ files, and that you're also collecting sample metadata, which made me think, could an extra carrot be that, hey, we're collecting all the stuff and then you can submit to GenBank? You don't have to fill out any of their forms anymore. It's just going to go in. There are certain places where that would be very enticing, I think, because sequencing a new genome, it's a new variety, it's a new subspecies, it's a new something. And it would be great to have that data shared to the community. But the biggest block is like, when you submit the data, they want metadata. And nobody wants to fill out metadata. But if they're filling it out already,
Right,
and if that's aligned with what GenBank wants, which I don't know if it is or not.
So if you have
would be
Aviti
cool.
users who are part of the AgBio consortium, I know David works with USDA previously. So data generated from an Aviti all contains a similar JSON file. So collaborators who are sequencing on Aviti would have a similar output file in their run directory that you would be able to collaborate with them and retrieve that JSON file that has the flow cell information, the run metadata that they provided during run setup. So you can imagine if you all had a CSV file that was shared amongst the community with similar project information, naming, you would have similar JSON files that could have a similar structure.
I think so. We have that wrapper that's the Amazon S3 is called Catalyst for us. Right now, we have a premium version that has analysis capabilities. It's on whole exome and whole genome analysis, but I could imagine that being feedback that we can bring back to our, kind of the marketing and product folks, where maybe in the basic version of Catalyst, they work to have some kind of export functionality direct to GenBank. Because I think that's right. Like, I think what Justin's saying is like, it's there for you to at least populate in the current state of things. But I think that's super useful feedback. Everybody does want to ultimately go into GenBank. And so that's And so or at least have an export functionality that it's going to be spit out into whatever format GenBank wants. So that's something we'll definitely bring back. And that's why we love, having these conversations. And Element's small enough still that it gets listened to pretty often, which is nice. We're in a sweet spot.
I've got a question. Sorry, it's Fringy again. I was thinking about the data file formats. You think of BAM, you think of VCFs. With the customers you talk to and with everybody here, do we think there's a data format missing that we should invent?
I mean, I was going to say most of my folks are FastQ VCF folks, but
So something that's been happening in the spatial field that I've been tracking is there's been a move to a spatial annotation data. And I wonder if something sequencing NGS side is needed as well that tracks the metadata from sequencing projects like that annotation data that goes with the spatial multiomics data set formats.
Sorry, this is Olivia. As someone who is kind of new to these file formats and is trying to work in machine learning to kind of have a standard format with columns that are informative and easily pulled from the data, I would have to say, please, dear God, no. Unless there is a very robust, standardized way that the files are formatted across a large corpus, like the AgBioData consortium. Otherwise, I feel like it could also add to the noise that's out there in some sense. Do you know what I mean? Just 2 cents.
You're not old and gray enough yet to be grumpy like that, but yeah, I had a question that was sort of similar to Tanya's, I guess, which was about like the biosample data that you're collecting, if it was roughly, equivalent to the core biosample data that, NCBI and those guys. I mean, I don't know, like, when you develop these standards and these formats, how much you were relying upon existing biosample data formats, or did you just invent something?
I'll have to check with our software team of what they had in mind when they envisioned the structure. I imagine they wanted to make it easy for GenBank submission, for SRA submission, for finding the metadata, trafficking the sample provenance. I'll let Amy add in.
The stab that I would take is they wanted it to not be far from Illumina in any respect, because that is what pretty much everybody was using when we first emerged. So my guess is it had to, not deviate from that. And then we really haven't put out any kind of new data type. So it was really staying within what was already out in the ecosystem. And we're following the same notes on the spatial side. So we're not putting out anything, you're getting count matrices and spatial data files that pop into existing standard spatial tools that can go into our pipelines, things like that. So, Element hasn't created anything unique on the data output side.
We just put a paper about markers for SNPs. What kind of identifiers are you using, like standard?
So we don't make SNPs ourselves. Is that what you're asking?
Just in your file formats, in your BCFs, what kind of identifiers do you use? Standard identifiers, like we promote the RSIDs. Yeah.
files. So the SCF files would come downstream through your analysis pipelines. So we produce the base calls, and then our base calling software, basis to FastQ, will take those base calls and work with the FastQs.
So you do positions.
Yeah.
Hello? Yeah, okay. Hi, I'm Heidi. I'm from the University of Illinois at Urbana-Champaign. First, just more love for provenance metadata in a JSON file. So however it ends up, it's very exciting, the idea that people wouldn't have to write in 50 cows, that's super. I had a kind of like an odd question that I wondered when you were talking about the just the decrease in cost, and it was mostly due to a decrease in reagents. So of the remaining cost, is it still reagent, or is there something else has superseded in terms of what the cost really is built up then? In?
Reagent, R&D too, right? There's some very fancy chemists who are figuring out how to make polymerases go faster and be better and all of that. So I think there's a standing level of R&D costs that's baked into any product that comes out in the market. And then there's the cost of consumption. We actually produce it. We're produced entirely in the United States. And so that, means we haven't been hit by a lot of the tariff issues and things like that. It makes it challenging when I try to do anything in Canada. But for here, that ends up being a net positive in terms of not having to pass any of those costs along. And the other thing, we didn't mention this, but for the Aviti and the Aviti 24, the NGS reagents, we actually, we're the only company that I think has ever done this, put out a price guarantee. So those reagent costs are guaranteed for the life of the platform. And so your cost today isn't going to have a 3% inflation year over year change. It's only going to get better if you do more volume and get discounts with that. So
yeah. Cool, thanks.